I'd like to tell you a little bit about work in progress that is ongoing in Utrecht. So I still feel a little bit modest because after all these tremendous talks that we hear here at the London Calling, I feel we need to be a little bit modest about our results. It's like I said, still work in progress. I still feel like a new kid in town, and hopefully I can take you through the steps that we have taken in Utrecht so far. So this is the newbie's perspective on needs. I have no conflicts of interest to disclose, so in that sense I'm okay. First, I want to show you and share you what we're doing here in Utrecht. We are at the center of the Netherlands, in the city of Utrecht. In the picture you see the UMC building and the Children's Hospital. We are part of the division Laboratory of Pharmacy and Biomedical Genetics, and we are basically a standard genome diagnostics center. We are working under an ISO accreditation, the ISO accreditation 15189. So all the tests that we are doing are accredited by the Dutch Accreditation Council. We have already a long history of next-generation sequencing. We're using that for all our diagnostics so far, and we have transitions from a targeted capture custom design assay all the way through whole exome sequencing and all the adaptations that were needed and asked for from the clinic. And for the coming weeks, we are actually transitioning to a short-read whole genome approach for all the gene panels that we are using in our diagnostic setting. We serve roughly 2 million people in our care area, and that is shared with all the academic centers in the Netherlands, so that are 8, so we are coming close to 16, 17 million people in the Netherlands. The aim is to build a robust and mature, high-throughput, long-read sequencing whole genome workflow. And we envision 4 different modules that we are actually developing right now. The first module is already existing because that's also kind of a module that we're using in our routine standard of care. And that is used for quantification and normalization. The biggest difference in this module is that we use a shaker for shearing the DNA to such a length that we can use it in nanopore sequencing. And for that, we use the FastPrep 96 that is developed by MP Biomedical. And for QCing the fragment length, we use the femtopulse from Agilent. The second module is actually quite new. It's still in an alpha testing phase. It's a bench-top machine for library preparation. It's called the Callisto, and it's developed by Volta Labs. It uses electrowetting technology for making these libraries, so there's no pipetting almost involved. It runs 24 samples on their flow cells, and it's kind of a plug-and-play idea, fully walk-away, so almost no manual interventions needed. And roughly in 2 to 3 hours, we have 24 libraries prepared. The third module, of course, is the sequencer itself. For that, we use a PromethION sequencer 24 system. And we are planning to use epitomy, of course, for the secondary analysis. And the fourth is kind of a gray zone for us still. There are some ONT development tools and, of course, third-party commercial software suppliers that are our preferred partners from ONT, and we're currently discussing with them to get them up to a standard that we can actually use it in a clinical space. We set a couple of preconditions when we started this pilot project, and that is the following conditions that we basically demand or want. We aim for a 25 to 30x long-read whole genome using a single flow cell, with the exceptions that we don't want to use any washing in between or reloading libraries in between the 72-hour sequencing run. That is different than most other groups are currently using. And the main reason for that is the ISO accreditation demands us to think in risks. And one of the risks of having a wash or a reloading step is actually mistakes either by humans or sample swaps or other things. So we want to avoid that. We don't want to use washing or reloading in our workflow. The fragment length that we're aiming for is an M50 score of about 15 to 20 kb. I think that is challenging, and I'll show you the results in a few moments. And we aim for at least 90 gig of data, and also that is challenging, as I will show you in the next slide. This is the learning curve that we have experienced in the lab so far. It's kind of a bumpy ride. This graph depicts the throughput versus the M50. So the blue bars is the output of the flow cell. The yellow bar depicts the M50 scores in fragment length, so in kbs. And you can see that it's actually quite bumpy, and it doesn't really give us a kind of a stable solution. And also what you can see if you look carefully at the yellow bars and the blue bars is that the longer the M50 score is, so let's say above 50, the less data that we produce on a flow cell. And remember, this is without any washing or reloading. I, of course, acknowledge that if we would implement washing and reloading, that the graph would look a lot different. And the other thing is that what we think we see in this slide, and also on the next slide, what we think we're seeing is also a shelf life effect. Although maybe in this picture, in this graph, it's not really showing that. But we have rerun some libraries that were producing not enough data on the older flow cells and rerun those on the fresh flow cells. And there you can actually see that the amount of data is substantially better with the fresh flow cells. So there is a shelf life effect, I think, so we need to think about stocking flow cells or the frequency of the supply chain. So where are we now? I think we are still capable of getting to a goal of 90 gigs using a single flow cell with no wash and or reloading. I was especially pleased with the presentation Rosemary gave yesterday, where she announced that using different buffer chemistry, that we would gain roughly 30% extra data. And even a 200 gig flow cell was on the horizon. So that would put us in a much better position if you look at the demands that we set in the beginning. 15 to 20 kb is challenging, I think, with an M50 score of that. But it's still a long-term goal, because I think that is actually really beneficial for our diagnostic workflow. For now, we will settle for 10 kb or more. And the main reason for that is that our bioinformaticians have shown that we do not really lose any or see any large effects on the number of haploblocks and the sizes of these haploblocks. But the next thing is probably the most important part, and that is that in a clinical setting, robustness of the flow workflow is actually really important. So there's a need for a surefire test that typically give the result that we want in a really stable manner. And that would probably mean less impact of shelf life, but it also would probably mean that we need a warranty on output. So we learned quite a lot from the first 100 flow cells, and we now move towards a more routine testing in our of clinical samples in our Nanopore flow cells. But for that, we need an automated library prep on the Callisto, and that is still in an alpha testing phase. But hopefully next year, I can present some actual data on how the Nanopore sequencing is actually detecting our clinical pathogenic mutations. And hopefully, I can show you some good results of the workflow itself. So with that, I want to acknowledge the people that are involved in this project, particularly Bert van der Zwaag and Koen van Gassen, who are really the drivers of this project. Of course, the technicians involved and the bioinformatics group involved, especially Martin Elvering, because he is already a long-time fan of Nanopore sequencing.